Lastly, I'm going to share with you a preview of our new software application called Biomek Method Launcher. And it's kind of a segue from the Rubicon method, because the Rubicon throughplex plasma-seq method is the first full method to have all the features of Biomek Method Launcher included in it. So what you see here is the Biomek Method Launcher home screen. And the software is basically it's an add-on to our existing Biomek software that's going to allow a simpler user experience when executing runs. So if you're familiar with the traditional Biomek software, you load it up and you see all the palettes and tools and different things that you can do with the method. And if you're looking at a complex method such as RNA access or Rubicon, you're going to see a whole slew of steps, and it can be cumbersome to somebody who is unfamiliar with the software when all they really need to do is push the play button and go. So what we've done is simplify that execution experience for them by creating a software layer over our Biomek application called Biomek Method Launcher. So the home screen is basically a tile-based application presentation. It's a one-click method execution for new or inexperienced users, and ensures that only approved methods are going to be presented to the user to execute. So it'll prevent an outdated version from being used. It'll prevent changes being made to a method that shouldn't be. Those applications can be organized however the lab wants. They can be organized by work group or process or what have you. And there'll be tiles and different icon buttons across the home screen. In addition, there's manual control interaction that can be accessed via the Biomek Method Launcher for homing or general pod movements, things that the user would still need to interact with the hardware. Usually you're purging the system or doing a homing before you start a run to make sure the fluid lines are filled, and that can still be executed without having to open up the full Biomek application. So one of the great benefits of Biomek Method Launcher is what we call guided labware setup. And if you're familiar with our existing instrument setup, it's basically a flat two-dimensional representation of the deck, and it can be a little bit difficult to know what labware is where. The names doesn't have hover over capability to hover your mouse over the plate and then have it indicate what the plate name is or what the labware type is. That can be done within Biomek software before you execute the run, but not at the actual instrument prompt when the method is started. So this takes that instrument setup process and completely overhauls it and allows complete flexibility of the person developing the method to define how labware gets placed on the deck. So in this particular example, we have three groups. We have a tips and accessories group, we have a reagents group with some specific labware that's listed, and we also have then a third group of sample labware. So each group will be executed in sequence. You just click through the next button, and it'll take you step by step through those groups and then through each piece of labware if there's specific instructions within a group for how to handle filling a plate or something of that nature. So in addition, it's going to have a higher resolution and clarity around stacked labware. So in the circled box here, there's actually a stack of plates, and you can see that represented in the table with the stack depth number, and also over here to the right, any position that's clicked on this deck view will have a profile view of the stack of labware. So before, where you saw three reaction plates stacked on top of each other, you had really no idea that there was three. It looked like a single plate. So now you have that resolution. You know the specific name and the labware type of each of those plates. And this can be customized depending on the application. There's going to be different ways to set up a deck depending on the application needs. So after the user gets through the guided labware setup, places their labware on the deck. Actually, there's one more step here. This is part of the guided labware setup. It's going to be the step that you'll see if you click into a specific piece of labware that requires additional work. So in the example, we have our reagent 2 block, and this is where we would traditionally see our reagent calculator with how to prepare your master mix or what wells to fill. That is now wrapped all up into guided labware setup. There will be specific reagent master mix preparation instructions based on the user inputs, how many samples you process, what part of the method you want it to run. And those instructions are provided up here in the green and red boxes. And then down in the well representation, it will tell you the specific volume and the specific master mix to add to each well. So it's full featured. It will guide the user exactly where to place reagents, prevent errors from calculating the master mix volumes themselves, and be very clear as to which wells those reagents are supposed to go into. And then once the plate is filled, then it'll instruct the user with the next tab where to place the labware on the deck. So after the deck is set up and the user's ready to run, then they are presented with the actual method launcher live view. So the live view is comprised of 4 primary tiles. Each of those can be popped out. There's a little arrow here, and you can expand that view to full screen and get a little bit more real estate on the information that's being presented for each one of those and toggle back and forth between them. So the top left tile is the milestones tile, and that can be configured however the application scientist wants. So in this case, there's a setup milestone, there's a library preparation milestone, and then there's the Ampere XP cleanup milestone. And what it'll do is it'll update in real time throughout the run to instruct the user on the progress of each of the steps of the run. As each is completed, a checkbox will appear on each milestone, and it'll continue and advance to the next milestone, indicating any subtasks of that milestone that are in process at that point in time. The method parameters tile indicates relevant method parameter selection information based on your user interface options. So if someone started up the run, selected 48 samples, and wasn't sure if that's really what they picked, and we're concerned that it was going to process 24 instead, it's going to show them that information. It'll show them all the options they selected around what method sections, index transfer options, any type of option that's available in the method that's relevant for the user to see during the run will now show up in the live view of the run. The Biomek deck tile is interactive. As I mentioned, the previous instrument setup was a flat view. You couldn't interact with it at all. Now this view has hover over capability. So if you hover over any of these positions, it's going to pop up the labware type and the labware name if it has a specified name. And those plates will move and be represented where they actually are in the method at that current time. In the labware view, it monitors all labware and well properties. So you're going to be able to expand the view of each of these pieces of labware and get a higher resolution as to what's in that plate. So Zach mentioned data sets and tracking index values from a source plate to a destination plate. You would be able to expand, oops, I'm clicking it as if it's live and it's not. You'd be able to expand the plate there and be able to see which indices are in each well and the types of data that you're associating with that sample. Volume is a default data set, but any type of sample ID information or barcode information that you're tracking through your process will be visible at runtime now and will update dynamically at each step in the method. So given that I just clicked on this because I want to show you how it works and it's just a slideshow, I really request that you come and check out the software in our booth because slides just don't show the type of interaction that you're going to get with this method launcher application. So we have 2 systems demoing it, and lastly, this is the kicker, is the ability to have web-enabled live run monitoring. So we actually have a system that's in our booth that's monitoring 3 other systems in their Biomek method launcher application. So it can be viewed from any portable device or PC, desktop PC that's web enabled. So it's pretty much everybody's phones now. So you can launch the application and have the same runtime view that I just showed you available on your tablet, on your smartphone, or desktop PC. So you don't have to be in the lab now and check on it every 30 minutes, pop back in to make sure things are going right. You can check it at your desk or go out to lunch and know that your run is executing properly. Okay, and I think our booth is 507. If I'm incorrect on that, then we should know now. But please stop by and check out our Biomek method launcher application in process and see all the features that it has, because it's really a lot more impressive than what we can show you in a few slides. Okay, and I think that's it. So this is just a slide indicating all the NGS applications that we have. We have brochures in our booth that you can take with you that are going to showcase all the applications and the platforms that they're associated with. So make sure you check that out. And again, thanks to all our collaborators from Rubicon, Illumina, and New England Biolabs for their help with our methods.